my fellow scientists. I have a question for you today. When you're hungry, how do you feel? I personally feel sluggish, maybe a little bit dizzy, maybe a little bit hangry, but most of all, I feel weak when I'm hungry. If you're like me, we feel weak when we're hungry because food serves as our source of energy. Feeling weak is actually our body's way of telling us that our energy supplies are too low. So, how does food get converted into a usable form of energy? We're going to answer that question in this lesson, which is going to focus on cellular respiration. So, grab your note packet, a pen or pencil, and let's get started. So remember that organisms get the energy that they need from the food that they eat. Cells break down food molecules gradually, capturing a little bit of chemical energy every single step of the way. This allows cells to use the energy stored in foods like glucose to synthesize compounds like ATP that directly power all of the activities of our cells. This process is called cellular respiration, and that's what we're going to talk about today. And once again, cellular respiration is the process of energy conversion that releases energy from food in the presence of oxygen. And we're going to see this in greater detail with the equation on the next slide. So as you can see, cellular respiration requires oxygen and a food molecule like glucose, and it actually gives off carbon dioxide, water, and energy. You can also notice that the cellular respiration equation is the exact opposite of the photosynthesis equation you learned about in the last lesson. Photosynthesis is the process that, quote unquote, deposits energy, while respiration, quote unquote, withdraws that energy. And what I mean by that is that photosynthesis removes carbon dioxide from our atmosphere, but cellular respiration puts that carbon dioxide back. Similarly, photosynthesis releases oxygen into the atmosphere, and cellular respiration uses that oxygen to release energy from food. So there are 4 main stages in cellular respiration, and some of them require oxygen and others don't. However, most of the energy releasing pathways that exist in our cells do require oxygen, which is the reason that we actually need to breathe. Stages of cellular respiration that require oxygen are called aerobic. So an example of that is the Krebs cycle and the electron transport chain. Those are both aerobic processes. And I know that these words sound a little crazy to you, but we're going to dive into those steps in a little bit. Any stage of the cellular respiration process that does not require oxygen, on the other hand, is said to be anaerobic. Remember that prefix "an" means not. So glycolysis is a really good example of this anaerobic process. However, we still include glycolysis in respiration because, as you'll see in a little bit, the final products of glycolysis are the key reactants for the aerobic stages. So without glycolysis, we wouldn't even be able to begin the Krebs cycle and have cellular respiration happen. So what is aerobic respiration? This goes back to cellular respiration, which we said at the very beginning on our first slide that it's the process that releases energy from food in the presence of oxygen. So, AKA, it's turning glucose into ATP. So glucose is our sugar, that's our food, and it's turning it into energy, which is ATP. This happens in the cells of all living things, and our cells are constantly undergoing cellular respiration all the time. And this process occurs in our mitochondria, which is why, as you probably remember from middle school, we always say that the mitochondria is the powerhouse of our cell. It is the area where our energy is being created. So a little bit of a summary of aerobic cellular respiration. Remember that we need glucose and oxygen to go into our system. Cellular respiration occurs, and 36 ATP and water come out of our system. So remember that our equation is 6O2, so 6 oxygen, plus C6H12O6, which is our formula for glucose, goes in, And what comes out is 6 CO2, so 6 carbon dioxide, 6 H2O, 6 water, and 36 ATP, which is our form of energy. Another thing to remember as a general summary is that there are 4 stages within cellular respiration, and those 4 stages are listed on your screen. Those stages are glycolysis, and then the Krebs cycle, and then the electron transport chain, and then finally our ATP synthase. So let's explore those stages in greater depth. So the first set of reactions in cellular respiration is known as glycolysis, which literally means sugar breaking. So during glycolysis, one molecule of glucose is broken down into pyruvic acid and releases a tiny amount of ATP, which is our form of energy. So once again, our input is going to be glucose, C6H12O6. Our output is going to be pyruvic acid, NADH, and ATP. And glycolysis occurs in the cytoplasm, which is right outside of the mitochondria. So let's deep dive into this stage. So even though glycolysis is an energy releasing process, the cell needs to put in a little bit of energy to get things going. That's why we see the 2 ATP up at the beginning of our pathway. Even though we are using 2 ATP, the process of glycolysis actually creates 4 ATP, which we can see here at the end of our pathway. So we have a net gain of 2 ATP molecules for each molecule of glucose that enters into glycolysis. On the right-hand side of our pathway, one of the reactions of glycolysis removes 4 electrons and passes them to an electron carrier called NAD+. Like NADP+ in photosynthesis, each NAD+ molecule accepts a pair of electrons and a hydrogen ion. This creates the molecule NADH. NADH holds the electrons until they can be used during the electron transport chain stage. Although the energy yield from glycolysis is small, remember just 2 ATP molecules, the process is so fast that cells can produce thousands of ATP molecules in just a few milliseconds. This can be a big advantage when the energy demands of a cell suddenly increases. Another advantage of glycolysis is that the process does not require any oxygen. This means that glycolysis can supply chemical energy when oxygen isn't present. When oxygen is present, NADH and pyruvic acid that is generated becomes the input for the Krebs cycle, which is our next stage. So in the presence of oxygen, the pyruvic acid produced in glycolysis passes to the second stage of cellular respiration, which we call the Krebs cycle. During the Krebs cycle, pyruvic acid is broken down into carbon dioxide in a series of energy-extracting reactions. So our input is going to be our pyruvic acid, our output is going to be our carbon dioxide, ATP, and NADH. And the Krebs cycle occurs in the mitochondrial matrix, which is the innermost compartment of the mitochondrion. Because citric acid is the first compound created during this stage, we sometimes call the Krebs cycle the citric acid cycle. So let's dive deep into our Krebs cycle. So the Krebs cycle, which you can see on page 317 in your textbook, and I highly recommend that you follow along on your textbook, it begins with our pyruvic acid up top. This pyruvic acid is produced by glycolysis, and it passes through our 2 membranes of the mitochondrion and into the mitochondrion matrix. Remember that the matrix is the innermost compartment of our mitochondrion. Once in the matrix, 1 carbon atom from pyruvic acid is split off to produce CO2, which is eventually released into the air. The other 2 carbon atoms from pyruvic acid is rearranged to form acetic acid. As the Krebs cycle begins, this 2 carbon molecule that you can see right here joins the 4 carbon molecule already present in our Krebs cycle, and this produces a 6 carbon molecule called citric acid. And that's this group of 6 carbon molecules right at the start of our cycle. So one of the carbon atoms in the 6-carbon citric acid molecule is removed. So here it gets removed, producing CO2. This occurs twice, so once right up top and then a second time a little further in our Krebs cycle, which then leaves us with a 4-carbon molecule that is moved again through this cycle. And that's why we call it a Krebs cycle. So now let's look at ATP. For each turn of the cycle, a molecule of ADP is converted into ATP. Remember that glycolysis produces 2 molecules of pyruvic acid from 1 molecule of glucose, so each starting molecule of glucose results in 2 complete turns of the Krebs cycle, so we get 2 ATP molecules. Finally, let's look at the electron carriers. Remember that we have NAD+, and we also have FAD as our electron carriers. At 5 places in the cycle, we've got 1, 2, 3, 4, and 5 right here. Electron carriers accept a pair of electrons and a hydrogen ion, changing NAD+ to NADH and FAD to FADH2. FAD and FADH2 are molecules similar to NAD+ and NADH, respectively. Remember that the products of the Krebs cycle are carbon dioxide, ATP, and electron carriers. The carbon dioxide actually diffuses out of our cells, enters our bloodstream, and then we actually exhale that carbon dioxide to release it back into the air. The ATP molecules then become immediately available to power our next stage. Finally, our electron carriers hold electrons that are going to be used to generate huge amounts of ATP in our next stages. So the products from the Krebs cycle and glycolysis feed into stages 3 and 4, which we said are the electron transport chain and our ATP synthase. The NADH created during glycolysis enters the mitochondria and joins NADH and FADH, which were created during the Krebs cycle. The electron transport chain uses the electrons from glycolysis and the Krebs cycle to synthesize ATP from ADP. So remember, our input is NADH. Our output for the electron chain portion is water. And the electron chain portion of our last 2 stages occurs in the inner mitochondrial membrane. So NADH and FADH2 pass their electrons to the electron transport chain, which is composed of a series of electron carriers located in the inner membrane of the mitochondria. During this electron transport chain, an enzyme combines these electrons with hydrogen ions and oxygen in order to form water. Notice that oxygen is the final electron acceptor. It's the final thing that is accepting electrons in our diagram, which is why electron transport is an aerobic process. So it requires oxygen because you need that oxygen in the diagram in order for the electron transport chain process to work. Without oxygen, this process wouldn't work. So every time 2 electrons pass down the electron transport chain, their energy is used to transport hydrogen ions across our membrane. During this process, hydrogen ions, which we can see up top up here, they build up in our intermembrane space, which makes it positively charged compared to our matrix, which is negatively charged. So our membrane essentially becomes a battery where electricity is kind of flowing, a charge is flowing, and that can power our final stage, which is the actual synthesis of ATP. So during ATP synthase, hydrogen ions move from a high concentration to a low concentration, adding a phosphate to ADP. So our input is our proton gradient, our output is ATP, and ATP synthase occurs in our inner mitochondrial membrane. So the cell uses a process called chemiosmosis to produce ATP. The inner mitochondrial membrane contains enzymes known as ATP synthase. The charge difference across the membrane forces hydrogen ions through the channels in these enzymes, actually causing the ATP synthase to spin. And with each rotation, the enzyme grabs an ADP molecule and attaches a phosphate group, which produces ATP. Every time a pair of high-energy electrons moves down the electron transport chain, the energy is used to move hydrogen ions across our membrane. These ions then rush back across the membrane with enough force to spin the ATP synthase and generate a huge amount of ATP. Each pair of high-energy electrons that moves down the full length of the electron transport chain provides enough energy to produce 3 molecules of ATP that can be used to power our cells' activities. So let's count up our totals for cellular respiration. So remember that glycolysis nets 2 ATP molecules per molecule of glucose. When you add oxygen to the mix, glycolysis, the Krebs cycle, the electron transport chain, and ATP synthase releases about 36 molecules of ATP per molecule of glucose. The Krebs cycle nets about 2 molecules of ATP, and the electron transport chain and ATP synthase together nets about 32 ATP. So 32 plus 2 plus 2 is 36. So as a summary, glucose and oxygen go into our system, cellular respiration occurs, and 36 ATP and water come out. The 36 ATP molecules that are generated through our 4 stages actually represents about 36% of the total energy of glucose. So what happens to the other 64%? It's actually released as heat, which is one of the reasons why our body feels warmer after exercise and why our body temperature stays pretty constant during the day. We are air-breathing organisms who use oxygen to release chemical energy from the foods we eat. So what happens if oxygen is not available? This is when anaerobic respiration kicks in. Cells and all living things undergo anaerobic respiration, which is a process that releases energy from food in the absence of oxygen. This process occurs in the cytoplasm of our cells. There are 2 types of anaerobic respiration, and both must undergo glycolysis to get our process started. The first type is alcoholic fermentation, which is carried out by yeast, and it produces ethyl alcohol and carbon dioxide. The second type is lactic acid fermentation, which is a process where a chemical reaction converts pyruvic acid to lactic acid. It's important to note that lactic acid fermentation does not give off carbon dioxide, unlike alcoholic fermentation. Alcoholic fermentation is used to produce beer, wine, and other alcoholic beverages. It's also the process that causes bread dough to rise. Since the yeast very quickly uses up any oxygen dissolved in the dough, they switch over to fermentation, giving off tiny bubbles of carbon dioxide. These bubbles form the air spaces that you see in a slice of bread. The small amount of alcohol that is produced in the dough evaporates when the bread is baked. Certain bacteria that produce lactic acid as a waste product during fermentation are very important to our food industry. So fermentation by these bacteria help to produce cheese, yogurt, buttermilk, sour cream, and basically all of your other sour foods. Lactic acid fermenting bacteria are also found in sourdough starters, and the acids they produce contribute to that very distinctive taste and texture of sourdough bread. Humans are also lactic acid fermenters. During brief periods without enough oxygen, many of the cells in our bodies produce ATP by lactic acid fermentation. The cells best adapted to doing that are our muscle cells, which often need very large supplies of ATP for rapid bursts of energy. This is also why you feel sore after exercising. It's lactic acid that is building up in your muscles. Well, scientists, that's it for cellular respiration. Now it's your turn to test your understanding of this material by working through the practice pages in your notes. Those are at the bottom of page 13 and all of page 14. Good luck!